All right, in this video we're going to talk about the idea of something called osmotic pressure. If you're a biology person, you've probably already heard about this, but I'm going to give you the slight chemistry approach to it. It is all based around the idea of something called a semi-permeable membrane. In most biological systems, it's something that allows water to pass through, but not any of the other stuff that's dissolved in there. Okay. For chemistry, the more broad definition is it allows solvents to pass through. So solvent molecules to pass through. But not solute molecules. You can almost visualize it like a filter. Okay? So we're talking about osmotic pressure. We also need to define what osmosis is. Osmosis is the idea that if there is a semi-permeable membrane separating two things that are different concentrations, the solvent will be allowed to move until the concentrations are the same. That's the entire concept here. And it creates, because there might be concentration differences, it creates a pressure by pushing the water one way or the other. Okay. So the solvent, osmosis is the solvent moving to make two concentrations the same across a permeable membrane. Two concentrations the same across a semi-permeable membrane. Okay. So let's kind of look at what this looks like. Okay. I'm not going to draw it from a biology perspective. I'm going to just draw it as a chemistry thing, but it applies to biological systems. Okay. So let's say we've got two containers, okay, and we have a semi-permeable membrane in the middle. I just drew it as a double line. I'm trying to oversimplify things. Semi-permeable membrane. Okay, so this little thing is a semi-permeable membrane. What that means is that it allows water to get through, but not, let's say, salt. So if on this side we have a low concentration, so we have some stuff dissolved, and you wouldn't actually see them because they'd be dissolved, but I'm going to draw the particles. Okay, this is a low concentration. And this side, I'm going to draw a lot more dots, would be a high concentration. All right, it's probably annoying to listen to. It's at least less annoying with this than with chalk. What we want to know is water is allowed to pass on this semi-permeable membrane, and because these two concentrations are the same, the water is going to move until these are the same. The question is, which way is the water going to move? Okay? Well, a lot of people, when they see this, they'll be like, oh, well, this is the lower concentration, so the salt needs to move over here. And that would be great, except the salt is the only thing that can't move through this. What we need to think of it instead is we have, relative to the amount of salt, a really high amount of water over here and a really low amount of water over here, because this is a high concentration. So the water will flow this way. It will dissolve the higher one. So let's say these weren't made out of glass. Let's say they were like, I don't know, plastic or something that can be squished and expanded. This side would shrivel up a little bit, and this side would expand to accommodate all the water until the concentrations were the same. Okay, and even if they were glass, the water would still flow this way until the concentrations were the same. Okay, but it creates an imbalance, which is why this is called osmotic pressure. There is inherently a pressure, the water pushing this way to try to equate everything, to make it all equal. Okay. So in the last video, we talked about the bends, which is a super morbid topic. I'm going to keep talking about morbid topics. Sorry, they're relevant to these examples. let's talk about fish. Okay, so osmotic pressure is the reason that it matters. Like if you ever have an aquarium, it's the reason it matters that you get the salt concentrations right. If you take a freshwater fish and throw it into salt water, it dies. If you take a saltwater fish and throw it into freshwater, it dies. Yes, I'm aware that there are some species that can actually bridge that gap, like salmon. Most can't. Okay, salmon actually have a way of like filtering out the salt concentrations to keep it all equivalent. Okay, it turns out that your cells are, for the most part, a semi-permeable membrane. If water needs to get through, it can. Okay, so if we took a freshwater fish, so freshwater fish in salt water. So let's say you bought a, I don't know my fish, goldfish, something that's a stereotypical freshwater fish. Okay, something you would get, it could survive in a lake, and you threw it in the Pacific Ocean or the Atlantic Ocean, whatever, some sort of really high concentration, or an aquarium designed for like sharks. Okay, what you would have is you would have essentially this situation up here. You would have low concentration inside the individual cells of the fish, and as it got exposed to that salt water, you would have high concentration on the outside. And the same trend we saw up here, water will flow from low concentration to high concentration. So what happens is the excess water inside of this will basically leave until the concentrations are the same. And if you have a really high concentration out here, this thing is going to shrivel up and die. Okay? So the individual cells will shrivel and die. Okay? Makes sense. I mean, it's essentially like if you want to dry fruit, if you don't care how salty it is, one thing you could do is literally just pour a bunch of salt on it, and it will pull all the moisture out, and it will shrivel up. And it's, I mean, if you don't mind your raisins getting salty, you could do this to grapes, and the salt would basically make it shrivel up like a raisin. Okay, this is what would happen to all the individual cells inside a freshwater fish if you put it into salt water. If you did the opposite, so let's say we have a saltwater fish, and you put it in freshwater. So again, I don't know my fish. I don't know, some sort of fish from the ocean, and you threw it into a lake. Okay, we would have the exact opposite situation. We would have a high concentration inside. The size of the circle has nothing to do with anything, I just can't draw. And you'd have a low concentration on the inside, or sorry, on the outside. So high concentration on the inside because it's salt water. It's used to having high concentrations. And as it got exposed to the low concentrate, the fresh water, it would have low concentration on the outside. Well, the idea is to make these two things equivalent, it needs to lower the high concentration. It's going to go from low to high, so all the water is going to rush into this. And the cell walls can only get so big. They're usually pretty full. As soon as water starts rushing in, it's going to expand, and it's literally going to pop, and it's going to explode the cells. So basically for this one, the cells will rupture and it will die. Yay! Either way is bad, don't do it. But what I'm going to tell you is that this is one of the reasons if you ever get an aquarium, paying really close attention to the salt concentrations matters a lot. Some fish can withstand a pretty broad range of salt concentration variance. Some can't. There are some fish that if you get this even just slightly wrong, one of these two options will happen and it'll die. Okay? But then on the opposite extreme, there's things like salmon. Salmon live in salt water, and they go upstream in rivers to lay their eggs. And then it gets washed back downstream after they grow up and eat, and they live in the ocean. So they basically make this happen. And they have ways of basically filtering out through their gills some of the salt concentration so that they don't have to expose the cells inside to high or low values. They can filter out and control the salt concentration in their blood as being independent and separate from what the concentration is outside of the fish. Kind of a weird concept, kind of a morbid topic because everybody dies in the story, but it's all based on this one really simple concept. Water will flow from the low concentration to the high concentration. And it's the water that flows, not the salt. And this happens all the time in biology because this is how cells are an example of semi-permeable membranes. Yeah, in biology you probably got a different spin on this, but this is the basic concept. All right, thanks.